Hello, my name is Nicole Arnold, and today I'd like to discuss my project, which included looking at differential gene expression in at-risk populations within Baltimore City through a cohort called HANDLES. And HANDLES is an acronym standing for Healthy Aging in Neighborhoods of Diversity Across a Lifespan. According to the National Institute of Health, any differences in incidence, prevalence, mortality, and burden of diseases are health disparities. Social determinants of health, such as healthcare, nutritious food, poverty, are all contributing factors to chronic inflammatory diseases such as kidney, hypertension, diabetes, and even mortality. My sample included African American and white male and females living above and below the 2004 poverty guidelines, which was the initial baseline of history and assessment. My workflow included obtaining peripheral blood mononuclear cells from each individual, which was then isolated for RNA that was used for a discovery microarray analysis and followed by a real-time quantitative PCR. My first figure is of the discovery microarray cohort, 1A and B representing African Americans, 1C and D representing white individuals within our cohort. And from here, we wanted to look and determine if gene patterns are altered as a result of poverty. And from here, we can see that yes, differential gene expression is occurring and associated with poverty. So the next step of the project was to go ahead and validate what we're seeing with the microarray cohort with a real-time quantitative PCR analysis. But first, we're going to start initially with just the African American males below poverty compared to above poverty, because previously in our lab we found that men below poverty had a higher rate of mortality when compared to other groups within our cohort. So go on to the validation cohort, we see that we were able to validate 11 genes that were up and down regulated within the African American male cohort. And of those 11 genes that were validated, 5 were statistically significant, differentially expressed.